So hello biology students, and welcome to video 2 of the biotechnology unit. So before we start in today's stuff, I'm going to give you a little reminder of what we did last time, just because it's a little complicated and I want to make sure we're all on the same page. So remember, we started with some piece of human DNA, and remember we had somewhere on that human DNA there was a gene we were interested in. And more specifically, we were interested in the gene for human growth hormone. Okay. So remember, what we did is we took a restriction enzyme and we cut up this DNA into a bunch of pieces. So now I have 1, 2, 3, 4, 5 pieces of DNA. And then I then took some plasmids from bacteria. Here's my 5 plasmids. I cut them with the same restriction enzyme, and then I stuck each piece of DNA I put inside a plasmid. That's kind of where we ended off the video last time. So now this kind of poses a problem, because now I have, 1, 2, 3, 4 plasmids that have DNA that I don't care about, and I have one plasmid right here that has my gene of interest. So one thing we're going to be looking at in this video is how to isolate only the plasmids we're interested in. Okay, so remember, we left off with the plasmids with DNA. So the next step is going to be to put my recombinant DNA into the host. So in this case, or in most cases, the host is a bacteria cell. So whenever you're putting foreign DNA into bacteria, this is called a transformation. That's a vocab word you should know. Okay. So in a transformation, recombinant DNA is going to be transferred to a host cell, and there are many ways you could do this. You could use what's called heat shocking, you can use electricity, or chemicals. But the thing that all these ways have in common is they all make the bacteria really stressed. So bacteria are kind of weird, and if they get really stressed out, they feel like, or the cell feels like it's going to die. So what it does is it basically opens itself up and takes in any DNA it could find anywhere. Bacteria do this naturally in the environment as well. And the idea is, the bacteria, it's programmed to think it's going to die soon, so it thinks by taking in foreign DNA, it just might find some DNA that will help it survive. So we utilize this idea. So we take, I have my bacteria, this is my bacterial cell, and then here's the plasmid I'm interested in getting inside of it. So if I cause this thing stress by heat shocking it or adding chemicals, it will take this plasmid inside of it, and now my DNA is inside the bacteria. The reason we're using bacteria is because they reproduce really quickly. So therefore, if I have my DNA inside a bacteria and it keeps reproducing, basically I'm going to have my DNA now inside lots of bacteria because they all reproduced. So this is a quick way for us to make many, many copies of that gene, or many, many copies of that protein, because they'll make my protein from the gene they have. Okay, so now we have this problem, because remember from the first slide, I had a bunch of plasmids that I've now put into a bunch of bacteria, but we don't know which bacteria actually has the plasmid I care about. Or some bacteria might have no plasmid at all. So maybe here's a bacteria, and maybe it has the plasmid, but this plasmid does not have my gene. Here's a bacteria with the plasmid, and this one does have my gene, so I'll put a little star on it. And maybe I have a bacteria that has no plasmid at all. Maybe it didn't take up a plasmid when I heat shocked it. So plasmids are designed in the lab, and that every single plasmid has a gene for antibiotic resistance on it. So in other words, any bacteria with this plasmid, if we expose it to antibiotics, they will not die because they have a gene to be resistant to antibiotics. So what we do is we take the bacteria and we basically grow them in the presence of antibiotics. And what this does, any bacteria that has no plasmid, they die. So that helps us to eliminate a lot of bacteria for whom the transformation didn't work. And now we're just left with bacteria that have a plasmid with our gene and bacteria with a plasmid without our gene. Okay, so now, at this point, all the bacteria we have that are alive all have a plasmid. Some of them have the plasmid we're interested in, and some of them have plasmids that we're not interested in. So all of these bacteria together, the idea is that in theory, if you looked at every gene, from the organism that you cut the genes from, all of those genes should be somewhere. So in other words, maybe if you had a chromosome, there's red pieces in this cell, and the yellow pieces in this cell, and the green pieces in this cell. So basically, together, they make up the entire chromosome. So we call all the bacteria together that have all the DNA, we call that a genomic library, okay? So a genomic library is basically kind of like, you can kind of think of how a library would have every book book. Well, a genomic library has a piece of every single piece of DNA from an organism somewhere in a bacterial cell. Okay, so it's kind of like a library of all the organism's genes. So now the next task is going to be to go through the genomic library and find the bacterium that has the gene we're interested in. Okay, so I'm going to go on a little side note here. Genomic libraries, a lot of them are going to have genes, but a lot of the pieces that are in a bacteria that we cut out with a restriction enzyme, they might code for nothing. They might not code for a gene at all. So remember we talked about way earlier in the year, when you have DNA, remember we said you have exons and you have introns? Remember those introns were the in-between sequences that didn't code for anything? Well, it's very possible that I have a bacteria that's just holding an intron, which is basically useless to me or useless to anyone because we can't really use that intron for anything. So a lot of times scientists will make what's called a cDNA library. Instead of making a regular genomic library, they make a library of just with genes that code for protein. They don't have any of the junk DNA. And they do this using this really cool enzyme called reverse transcriptase. So basically what this is it's an enzyme that takes all the mRNA from a cell. Remember the mRNA is a copy of the DNA that's actually going to be used to make proteins? So this enzyme takes mRNA and turns it into DNA. So it's called reverse transcriptase because it's doing the process in reverse. It's turning mRNA into DNA. So if we get our DNA from this process and then we use restriction enzymes on this, so I'm going to put a little RE for restriction enzymes. So I cut up that DNA and then put that into plasmids. Now I have a genomic library only with DNA that holds genes. There's no plasmids holding any junk DNA. So that's a lot more useful. It's more useful because it cuts down on the number of plasmids you have to work with, but it's mostly useful because bacterial mRNA actually has no introns. So if I put a plasmid with an intron into my bacterial cell and I want to make a human protein from that, I can't because bacteria have no way to process those introns. So it's really important that my eukaryotic DNA that I put into the bacteria is lacking in introns. Okay, so here is it's an example to show you that. So maybe here's the gene I'm interested in, but it has these introns, and the bacteria is not going to be able to deal with those. So I get a copy of the mRNA from the human cell. So here's my mRNA, and then I use this enzyme, reverse transcriptase, to make the DNA copy. And if you look, my DNA copy now has no introns. So if I put this piece into a plasmid, now the bacteria will actually be able to read this DNA and make the protein for me. Because remember, the whole point is to get the bacteria to make the protein for us. Okay, so now, finally, we're going to talk about, so if I have a plasmid that has a bacterial cell with a plasmid with my gene, and I have my bacterial cell with a plasmid that does not have my gene, how do I figure out, which one has the gene I care about? So we do this, we figure it out by using what's called a probe. So basically, we know DNA is made of nucleotides, right? So if somehow, here's my single-stranded DNA, and maybe it has some sequence on it, okay. If I could somehow separate the DNA, which we're able to do, so separate the two strands, and then build my own DNA that will bond to one of the strands, and if I'm able to find that piece of DNA that I made, we can figure out which cell has my plasmid, and that's exactly what we do. So we use what's called the probe, which is basically a radioactively labeled single-stranded DNA piece. So it's a piece of DNA that's been labeled with something radioactive, so if you put it under certain conditions, it will glow, so you can physically see it. So maybe, I know somewhere on my gene is the sequence TAGGCT. So I know in my gene, in my human growth hormone gene, there's this sequence. If I'm able to make it so all of the DNA and all the bacteria separate so they're no longer double-stranded, and then I add a single-stranded piece with this fragment, so I add this to the bacterial cells, and this single-stranded piece, this probe, has a couple nucleotides that are radioactive so they glow. If I could then look under a light, I can find my plasmid that's glowing, and I know that's the one that has my sequence, so that must be the one that has my gene of interest, and this is exactly what scientists do to locate the right bacterial cell. Okay, so here's that example. So maybe this is, a piece of the gene, of my gene of interest. So I separate it using usually heat, and then I add this probe to my bacterial cell, and then it's only going to bond to my gene of interest. So now I just find the cell that's glowing, and that's the one that has my, my gene. So here's another example of the same thing. So you can see here's all the bacteria. They all have plasmids because we use antibiotic resistance, but I don't know which one has the plasmid I care about. Okay, so we're going to add the probe to those plasmids, and then the ones that glow, those are the ones I know that have my gene. So I would then take those colonies that I care about and then grow them on their own and use them later on for further analysis. Okay, so one more time to summarize the whole process. So in restriction enzyme technology, we're going to take a plasmid, we're going to take our gene of interest, combine them, and then put it in the plasmid, allow it to multiply, find the one that has the plasmid I care about, and then now I have tons of copies of my gene, and these bacteria can make tons of copies of my protein, and then I could then use that human growth hormone protein to treat people with dwarfism. So that's it. See you in video 3.